Cells moving, meeting, and moving away again. Movement achieved by the ruffling of the cell membrane. This shows the membrane as a mobile and very dynamic structure, but a freeze-fracture image of the cell reveals a structured and rather static image of the membrane surface. And a conventional electron micrograph, here two membranes closely opposed, shows each to have a tram-like appearance, a trilaminar structure. Now, biochemistry tells us that the membrane is composed of proteins and lipids. Can we construct lipids and proteins into an arrangement that will fit with these images? Whatever arrangement we construct must take into account the functions of membranes. Membranes have to be permeable to allow nutrients to enter the cell, and yet be impermeable to maintain the integrity of the cell. To solve this paradox, let's look at the arrangement of the principal macromolecules. Dr. Dennis Hayden, University of Cambridge.
Hydrocarbons don't dissolve in water. The water's coloured for the purposes of television. Now, phospholipids, for this reason, don't dissolve in water either. They form aggregates. In a biological membrane, these aggregates take the form of a bimolecular leaflet. That is a leaflet just 2 molecules thick. Just 2 molecules. Now, why 2 molecules? The answer is to be found in the structure of the phospholipid molecule. This molecule has a polar group and 2 non-polar chains. The polar group tends to position itself in the aqueous phase, and the non-polar chains tend to associate together so as to minimize their contacts with water. Now, if we look at the dimensions of the molecule, we find that the cross-sectional area of this polar group is very similar to the cross-sectional area of those 2 chains. This makes the molecule roughly cylindrical in shape, rather like this beer can. And it's because of this cylindrical shape that the molecules compact very easily side by side. The beer cans illustrate the point very well. Here we have a monolayer of phospholipids, polar groups and the chains. Now, as you see, the ends of the chains are still exposed to water, and so this structure is not ideal. However, if we put another monolayer back to back to the first one, so that the chains are in contact with each other, we can completely exclude contacts of the chains with water, and we have pretty well the ideal structure. In this structure, the chains fill all the available space, and in biological membranes at ambient temperatures, the chains are in a liquid state.
Now, just such an arrangement is likely to produce a significant barrier to the movement of small, useful substances, such as ions, amino acids, and sugars. We can study the permeability of these across membranes in a number of ways. Phospholipids in the presence of water naturally form lipid bilayers, such as this liposome. But when we look at the structure of the liposome, what we see is that it's not a single lipid bilayer, such as in a normal cell membrane, but a series of lipid bilayers arranged concentrically. The multimeric structure of the liposome makes it difficult for permeability studies. Now, the way out of this is to construct single lipid bilayers like black lipid membranes.
These black lipid films are formed by dissolving a phospholipid in a hydrocarbon solvent and extending this solution across a small hole in a suitable support underwater. It isn't easy to see on this, so I'll illustrate it in this diagram. This is the aqueous solution in this Perspex container And this is the Teflon support with a hole in it, across which we make the film. So when I make a film, it forms roughly like that. The phospholipid in the hydrocarbon is in this Pasteur pipette. And I just place this close to the hole and squeeze a little out. When the film's thick, we see characteristic interference colors in reflected white light. Okay. This black film is an extremely convenient means of measuring the permeability of a lipid bilayer. For example, it's easy to demonstrate that the bilayer constitutes a significant barrier to the permeability of water-soluble material. In the cell at the moment, we have a dye on the far side of the membrane and nothing on this side. That situation has been in existence for several hours and no dye is coming through the membrane. But break the membrane and watch what happens. So lipid bilayers are very impermeable to water-soluble substances. Now, the movement of inorganic ions, like sodium and potassium, in and out of cells can be very important. For example, in nerve impulse conduction and in muscle contraction. And we would like to know how to investigate what is responsible for these ion movements. Ions are electrically charged. So, when they move, they generate an electric current. This current can be measured if we put electrodes on either side of the membrane and apply a voltage between them. The current which flows is related to the permeability. Using Ohm's law, the membrane conductance, G, is given by the current divided by the voltage. The voltage is constant, the current is measured, and therefore we have the conductance. And the conductance gives us the permeability. So let's look at an experiment in which we examine the permeability of the bilayer to potassium ions. The traces on the oscilloscope show the upper trace, the voltage, which rises, and the lower trace, the current, which also rises, but not very much. We turn on the voltage, you see a big rise in the voltage, but a very small rise in the current. This current is in fact much smaller than you would see in a normal biological membrane under these conditions. We turn off the voltage, and the current goes back to normal. Now let's see what happens if we add to the membrane a certain type of polypeptide, gramicidin. The polypeptide will be added to the solution near the membrane and will become incorporated in it. Again, we have the two traces. We turn on the voltage. Initially, the membrane current is as it was before, quite small. But gradually, the polypeptide diffuses to the membrane, becomes incorporated in it, and the current rises faster and faster, disappears off the top of the screen, and reaches values which are comparable to or even greater than those which we find in a normal biological membrane. How can we explain this very large rise in current, and hence in membrane conductivity, that we find when we add the polypeptide? Gramicidin consists of 15 amino acids. These are coiled into the form of a helix, and in such a way as to leave a hole 4 angstroms in diameter down the middle. In the membrane, this hole is filled with water, and it's through this aqueous channel that the inorganic ions move. In point of fact, 1 gramicidin molecule isn't long enough to span the lipid bilayer, and 2 are needed. This is the dimeric gramicidin channel, hydrogen bonded together in the middle. These are sodium ions. An ion channel. When we add extremely small quantities of gramicidin to a bilayer membrane, apply a voltage, and watch the current, we see a very interesting effect. This is the baseline current corresponding to a pure lipid bilayer. Arising out of this, we see suddenly square pulses of current which correspond to the formation of 1, 2, 3 individual gramicidin channels. These events occur at random, and they correspond to the coming together of 2 monomeric gramicidin molecules to form the dimer. Now, the fact that this random formation of dimers can occur means that the bilayer must permit a free lateral diffusion of these molecules to and from each other. This in turn means that the bilayer is a fluid structure.
So ion channels, created by the insertion of polypeptides like gramicidin, change the permeability of the lipid bilayer, what occurs in living membranes. In order to determine whether it's the proteins that are concerned with permeability, we need to look at their arrangement in the membrane. The original idea of Dowson and Danieli had the lipid bilayer sandwiched between continuous smears of protein, but Singer and Nicholson had other ideas. Professor John Singer.
What we've learned about proteins has come largely from studies of X-ray crystallography of water-soluble proteins in a crystalline form. And there it's become clear that the molecules of proteins are globular, compact structures, very complex conformation, and not single alpha helices at all, but convoluted into some shape in which the hydrophobic amino acids are intercalated largely into the membrane interior, and the hydrophilic amino acids are largely on the membrane exterior. This maximizes simultaneously both the hydrophobic and the hydrophilic interactions. And the question was how to relate this kind of structural information to the problem of membrane proteins. Now, I entered this field about 1960. My background is a protein chemist. And the question was how to relate these problems to the problem of membrane protein structure. The solution was obvious to most protein chemists if they had addressed themselves to this problem, and that was to have a protein molecule organized as an amphipathic structure in which the hydrophilic amino acids would be largely localized to the exterior surfaces of the molecule where the protein would come into contact with the water, and have the hydrophobic amino acid residues localized to the interior part of the molecule where it would come into contact with the hydrophobic fatty acyl chains of the phospholipids. Now, what happens if we try to place a molecule of that structure into the lipid bilayer of the membrane? That can be visualized in this model. What you see resulting is that the hydrophobic surface of this amphipathic protein is in the interior region of the membrane where the hydrophobic fatty acids are located, whereas the hydrophilic exterior of the protein is placed out into the aqueous environment. And if this protein is one which spans the thickness of the membrane, then there are these two hydrophilic surfaces exposed at either side. Now, if we look at the top surface of this model, what is seen is that this represents a kind of mosaic structure in which the individual proteins are randomly disposed in the lipid bilayer. In particular, if the lipid itself is in a fluid condition, then there are no restrictions holding these proteins in any particular place in this plane, but rather they should be freely mobile in the plane. Now, this picture of membrane was quite unexpected. Most people who thought about membranes at the time thought of them as rigid structures in which the proteins formed some kind of a lattice which was quite immobile. But around this time, Fry and Edidin performed a very remarkable experiment. It is possible to attach fluorescent labels to the surface proteins of living cells. A label of one color was attached to human cells and of another color to mouse cells. The cells were then made to fuse together. Initially, the labels were separate, but within a half hour, they had completely intermixed. Another problem that this model addresses is the problem of permeability and transport. By a minor variation of the protein structure that we've been talking about, we can, for example, envision that a protein exists in the membrane as a kind of subunit aggregate with a water-filled channel running down the interior portion of the membrane through the membrane to the other side. The point here would be that the channel would be lined by hydrophilic residues of the protein molecule so as to satisfy the thermodynamic interactions involved. And the small molecule ion or other hydrophilic ligand could then traverse the membrane from one side to the other. One of the main reasons to have a protein being transmembrane, I think, is to be involved in these transport processes. The existence of proteins that span the entire thickness of the membrane is most aptly demonstrated by freeze-fracture experiments which have been pioneered by Daniel Branton of Harvard University.
Very often we can actually see transmembrane proteins by freeze-fracturing. For example, in the case of the red blood cell membrane, which we know is very active in anion transport, one can see a large number of bumps on the surface of the membrane, and in fact it's clear that these bumps do represent the transmembrane proteins that serve as anion channels. In contrast, on this photograph, for example, of myelin membranes, we see a relatively smooth surface. These faces are representative of membranes which are inactive in terms of transport. In fact, they serve primarily as insulators around myelin. But the differences aren't all that clear-cut. In many membranes, a protein traverses through the lipid bilayer as an alpha helix only once. And then it's too small to be visualized by freeze fracturing, so we see nothing. We don't see any bumps, even though there is a transmembrane proteins. Nonetheless, because transmembrane proteins frequently go through the membrane more than once, freeze fracturing serves as a very useful way of looking at these transmembrane proteins.
So far then, our model looks like this. Proteins lying in the lipid bilayer, floating freely in a lipid sea. But what's the importance of this fluidity? One of the key functions of membranes is to communicate events happening outside the cell through to the cell interior. And many of the proteins in the membrane act as receptors for signal molecules like hormones. And many hormones, in fact, exert their cellular effects by activating an enzyme, adenylate cyclase, which is in fact well buried into the membrane on the internal face. Activation of adenylate cyclase produces cyclic AMP from ATP, and it's cyclic AMP, this second messenger, which initiates the cellular response. Both proteins, receptor and adenylate cyclase, appear to float freely in opposite sides of the membrane. Random meeting elicits no response, and they drift apart. But when the hormone is present, and this binds to the receptor, it activates the receptor such that a subsequent meeting of the hormone receptor complex with adenylate cyclase now results in the production of cyclic AMP. But in the same way that fluidity permits the coming together of proteins in the membrane, it can also take them away from one another, and this would not be very satisfactory in terms of function. It seems, in fact, that not all parts of the membrane of a cell are equally fluid. There are areas in which fluidity is controlled, such as the junctions between cells, the so-called gap junctions, and the points of interaction between the cells of the nervous system, the synaptic junctions. Clearly there is some type of modulation of the membrane going on, but how is this modulation brought about?
At least in one case, we've been able to isolate the connecting proteins and examine their connections. In the red blood cell, for example, this model has emerged. We know that there are at least 4 major elements involved in connecting short actin oligomers to the membrane. These elements include a connecting protein called spectrin. Spectrin in turn binds to ankyrin, and ankyrin binds to band 3, which is the major transmembrane protein. We've been able to study this set of connections directly by purifying the proteins and examining them individually. For example, in the electron microscope, spectrin molecules appear as long, fibrous elements. We can incubate spectrin together with ankyrin and see directly in the electron microscope where the ankyrin molecule binds to the spectrin molecule.
We can see how these connecting proteins link together to form a network. In essence, then, a meshwork underlying the membrane regulates, modulates the movement of specific transmembrane proteins.